I've never been more excited to give a talk because I had to stand up. I thought I was going to have a DVT. So thank you for this opportunity, literally. So I'm going to start off with Frontline here and look at where we've been. And I think, we think about all the changes we've made and the slogan that we have, which is really our mission, is to transform the standard of care, as Dr. Copeland said earlier. We've really seen amazing data come out in frontline ovarian cancer, and I'm going to show you just sort of a couple slides on that to set the tone and to look at where we've been with maintenance. But it wasn't until really 5 years ago that started to happen, right? And if you think about up till 5 years ago, we were really struggling with the same thing since the original article came out in 1996, looking at the taxane era from GOG 111. And so I think it's really incumbent upon us to keep that in mind. We went down a lot of rabbit holes, if you will, dose dense therapy, IP, and we'll get back to HIPEC in a moment here. But the maintenance strategy certainly has been amazing, and we've started that with bevacizumab, with its approval, but then look at what we've done with PARPs, and the PARPs have just totally transformed those with a positive molecular signature, and certainly those that are HRD test negative. I think you could argue one way or the other on those patients, and we'll probably have that argument very shortly here. And this obviously informs the current recommendations based on NCCN of treatment in the frontline setting that I know you're all familiar with, and the supporting trials to back those up. What was interesting that came out at ASCO this year was a poster that looked at the PALO-1 study. And just to remind you, these are advanced stage 3-4 patients, high-grade serous, who then went on to get randomized to either olaparib plus bevacizumab or placebo plus bevacizumab. This analysis, conducted by Phil Harder et al., looked at the first progression and the second progression, generated curves and medians off of this data, and compared those patients that progressed on a PARP versus after a PARP versus those that were PARP naive, okay? And so here's the Kaplan-Meier curves, which is very interesting. So these are all progressors, and remember, about 2/3 of the patients eventually progressed in that trial. So keep that in mind. But here you look at this, and you see that these patients that were PARP naive did just as well as someone who progressed after a PARP compared to the group that progressed on a PARP, which is in the blue there. So those patients did significantly worse. This is supported by the medians that you see here that set that up. And then if we put this into context of looking at sensitivity analysis, you see progression after or during a PARP inhibitor, in this case olaparib, really was predictive of your outcomes. And so this is a post hoc exploratory analysis. Even in the conclusions, they said this data should be interpreted with caution. There's a lot of holes, in my opinion, with this data in terms of the analysis. This is one that I really would need to see the manuscript, because I think the reviewers are going to request more data than what was in the poster before we actually get it into peer review. It's a great idea, but it really questions whether this whole issue of whether we are perhaps even harming patients with PARP exposure, maybe not in the molecular positive group, but in the other group. Remember that even though the label was different, the trial itself set that up for this comparison, because we had HRD positive patients in here and those that were HRD test negative.
Yeah, thanks, Tom. And I am really looking forward to breaking this down. So this is, I'm going to go.
Oh, you're going back, okay.
Let's look at this. So first of all, this includes all patients on PALO-1, okay? So I don't want you guys to jump to conclusions and say, oh, the patients that didn't get a PARP inhibitor did just as well as the patients that did. This is the key thing here. The challenge is that suggests just what I said. And so if you look at the progression during olaparib, after olaparib, and the control arm, and this breaks it down according to the HRD positive subset, In the HRD positive subsets and the HRD negative subsets look similar. Now I'm going to go back a slide. So maybe this, although it is an all-comer population, suggests that you don't really need a PARP inhibitor. Now I'm going to ask the question I think that everyone wants to know. Is this just sorting out bad biology? Because if you progress on treatment, whether it's platinum, which really this is a surrogate for, or are we hurting patients with PARP inhibitors? Katie, I know you've thought a lot about this. Help us interpret this very provocative. And to your point, Tom, I'm not sure I really get it. I kind of need to read the paper.
Yeah.
But this is probably the most provocative abstract that was presented at ASCO.
Yeah, and it's exploratory, and we need the paper. 100% agree. So 2 points on this. So 2 things. One, the thing that surprised me was that there was no difference. So this is all the patients who recurred who got platinum, and then what happened? Their PFS to their next line of therapy. So it surprised me that the curves of the patients who didn't get any PARP on Paula and the patients who did get a PARP but progressed after completely overlay one another. I would have expected a gradation of response there. So your green and your red lines are completely on top of one another.
Right.
The blue line is kind of a no-brainer because they progressed on a PARP. And so that if you're progressing within 2 years on a PARP, that's a particularly aggressive tumor. You're on something active and you're progressing. But I do think the thing, to your point, Tom, that the crossover can't explain is look at the difference between the red curve, which is control, and the blue, which is the progress on a PARP. So the control arm, a lot of those patients progressed in 2 years too. So they're early progressors. They're all in that red line.
Yeah.
And they still, if you look at that, you say, well, it's just crossover, but they all got platinum, so that's the first 2 bars down there to 6 months. They're separated already. In that first 6 months, that patient group who progressed on a PARP is doing worse than the control group on platinum. And that concerns me. I'm not surprised by it, but it concerns me, and I need more data there. So I think that separation to me is the most concerning thing, and I want to see some more granularity. We won't see response rate because they didn't collect that, but that group concerns me.
So Ramez, we've always said that the highest unmet need in first line is the HRD test negative, and that's probably still true. But is it also now those that are HRD positive that progress on a PARP? Are there 2 high unmet medical needs in first line, HRD negative, and those that progress on PARP that are HRD positive? Go ahead, Dave. I see you shaking your head. Then Ramez, you have a comment.
I was going to say yes and yes. Why do I say this? We can't forget we have OS data from PAOLA-1.
Okay.
And the OS data in the HRD test positive suggests an
substantial.
advantage, numerical advantage to olaparib plus Bev. The key here, and this is where it gets nuanced, and I agree with all the comments, and the manuscript is important in granularity. If you have an HRD test positive patient and you use olaparib and Bev, and we know the advantage based on PAOLA-1 over Bev plus placebo, you have a dramatic benefit. Then you re-challenge at progression on PARP. Does the magnitude of benefit you achieve in the beginning abrogate any potential compromise down the line? We don't know. But in the biomarker negative, where we know that Bev plus placebo was no different than olaparib plus Bev, they overlapped each other completely. You don't have that advantage anymore. You can't play catch up if you have a biologically different tumor that progresses on PARP, to Dr. Moore's point. So that's where I think this is where the nuanced conversation happens about the biomarker negative progress on PARP who previously otherwise would have gotten Bev as a monotherapy.
Yeah, so Tom, thanks for introducing this to us.
Yeah.
Dave, if there are no comments, we'll move ahead.
I just want to say one last thing on
Yeah.
that, though, that one of the things we want to know is crossover.
Yep.
That first subsequent therapy for the control arm could have been a PARP.
Yep.
The other thing that is interesting is if we look at it within the label, how did those patients do? So if we look
Okay.
at just those with the molecular
Anyone with
signature.
one more comment? I'm sorry.
Yeah, it's fine.
So
Love it.
can you go back? So the other
Absolutely,
thing,
I
we
can
don't have time
go
to
back.
talk, but look at the poster, because the other thing that the poster did was it broke out the clinical pathologic features of the patients who progress on PARP versus not. So these are all the patients who progressed. They're not the patients who are cured, who are largely BRCA.
Correct.
So those are excluded. So this is mainly going to be bad performing BRCA and BRCA wild type.
Yeah.
So, and we need that clarification. But there is
It's
a
like
chart.
45% versus 28%.
Correct.
Yep.
Right, so it's very heavily weighted towards a different demographic or tumor classification and maybe not distributed appropriately. So exploratory, and we need the manuscript. This isn't the whole population.
And it's
The patients who
yeah.
are cured are not in here.
I love it.
And I just have one comment.
Thanks, Dave.
Is this is not that surprising. We've been talking about these. What is PARP resistance? Okay, PARP resistance, is it exposure or is it progressed on PARP? I think this shows that those that progress on PARP are definitely a different patient population
Yeah.
than those are exposed. I've heard people say this gives us more ammunition with regards to, oh, we should hold off on PARP. We're, in Palo, we cured like 45% of patients.
Rama said it well. There's survival
I mean,
advantage.
so
Yeah, there's a
you're
survival
not going to
advantage.
become platinum resistant if you never have to get platinum again.
My term. So Duo, that was easy, Tom. Go ahead and tell us how we
Yeah. It's not a typo, it's Dr. Harder again. So this is the Duo, which everyone anticipated. So there are 4 main trials that we were looking for in combinations with PARPs and BEV and so forth. And this is the first one to report out. So we have Key-Link 001, we have the Athena mono, or Athena combo rather, that'll report out. And then this is the first one that we have with Duo that's actually reported at ASCO. And here you see arm 1, which is chemo plus BEV, arm 2. So almost look at the left hand, it's a lot. When I looked at it, when they first presented it, was very confusing because you see all those placebo, they put the name and then placebo after it. But arm 1's just chemo plus BEV, arm 2's chemo, BEV, and durvalumab. And then chemo, BEV, durvalumab, and olaparib in arm 3, okay? Primary endpoint, PFS, and then step down there. So what is important is to understand how they did this analysis. What they did is they compared arm 2 to arm 1 and arm 3 to arm 1. They did not do a statistical comparison between arm 3 and 2, which many of you would have liked to have seen. Patient characteristics were fairly well balanced. You can see here those characteristics across the study. This is the non-tumor BRCA HRD positive group, arm 3 versus arm 1. Clearly, this arm was superior with a hazard ratio of 0.49. How much of that was driven by just the olaparib versus the durva being in there is the critical question for this entire study. Here's the PFS for the intent to treat population. As you can see here, hazard ratio was 0.87 compared to arm 1 for adding in the checkpoint inhibitor with BEV. And then the much more impressive when you add the olaparib in there. So you do see there's room between those curves. So some people could say, well, there is a benefit to adding the checkpoint inhibitor. However, the greatest benefit was clearly the triplet, but does it confirm that that's what was needed? Could you have dropped the checkpoint inhibitor and done just as well? So many of these arms, as we get into this discussion, you will see people will talk about the fact that there are missing arms here. And this is looking at by HRD status to try to better understand if there's a difference between these. Again, for the HRD negative, you really don't see much benefit there compared. So I just threw one safety slide in. There were several of them. I've hidden the other ones, but just to show you that it does incrementally increase as you add agents, not surprisingly. And that's all we really need to say about that. And then these are the 4 trials here. And the important thing is remember that you can take the Palo 1 curve, as some people may have done that aren't here today, who are usually here, Rob Coleman, and you can really see where that fits in as well. And it gets very interesting to do that. Obviously, unconventional and not to be encouraged to do cross-trial comparisons like that. But that's the missing arm of this particular trial that we're trying to interpret in terms of Duo. I think we're going to see some interesting things. With first reading out, I think first, where you look at just chemo, then niraparib, then chemo, niraparib, plus dostarlimab will be interesting. And then Key-Link 001, both of which are going to be based on a primary endpoint, which is going to include PD-L1,
Yes.
which I think from a technical chance of success, that probably improves that if indeed an IO is important in ovarian cancer. And then we can talk a little bit about how BEV fits in. BEV is optional in the first trial. And so that can taint this a little bit. Athena will be a little cleaner, but it could limit clinical, the ability to apply it into the clinic if you're a BEV user, because no BEV in that particular arm. So it'll be interesting to see how all this data comes out.
That's so much fun. I'm a little disappointed because I don't really feel satisfied here. So Katie, so if you look at arm 1, PC Bev, arm 2, PC Bev, didn't we already do that in Imagine 50?
Yeah. Yep. Negative.
Okay. Any comment?
Well, I mean, this was designed. This was
I get it.
designed
Yeah, we have
when
to
we
be
hadn't
fair.
had those results, and so you sort of guess and
Correct.
think where the field's going to be, and you take a risk and run a study. So it's easy to be an armchair quarterback and make fun of it. Although I did
making
did
fun
have
of
a
it.
stick figure with no arm when I presented this, so I did make fun of it. But that's not very nice because it was well designed by smart people, and it just is what it is. But it's unfortunately uninterpretable.
So Dave, don't we already know that olaparib, when added to Bev, makes it better in PALA-1? Isn't that what this shows?
Yeah, this is, I think, as I look at this and, cross-trial compare, which I don't do because that's not valid.
But I'm unsatisfied.
No, you're unsatisfied. This is, I think, a negative result. Okay, we're not adding Durva. We're not improving the outcomes in HRD positive patients. As I look at the HRD test negative patients, maybe there's something there, maybe, but not in HRD positive.
Yeah, we can
Okay,
tell you
well,
that.
first of all, Bhavna, how good would this have to be to use 3 agents in the maintenance phase?
Yeah, no, I mean,
But how good would it have to be?
it would have to beat,
Amazing.
it would have to be amazing.
And this isn't even,
With the toxicity of 3 agents, right?
0.5 if it was a real comparison. And
right.
if that was bevelaprib and we hit 0.5, I would absolutely use 3 agents. But
Nope.
this isn't a real 0.5.
That's right. It's
No,
not a
this
real.
is over just bad.
So much fun. So let's go to the HRD test negative here, right here. So it's interesting that the arm 1 versus arm 3 here, 0.68. Ramez, what was the hazard ratio on Prima HRD test
Almost the
negative?
same. It's the same.
Like almost
Exactly the same.
So 0.68 is pretty
0.68.
close.
Against placebo, against placebo.
So, but that was in both arms, so it cancels out.
And you're still so this is what, but in PALA-1, you had no advantage, maybe, if anything, I'm not going to say detriment, but definitely no advantage of adding the olaparib to Bev in HRD test negative patients.
It's a non-analytic subgroup.
It's a
And
non-
this is a non-analytic subgroup.
Yeah, this
You
is
don't know which one.
non-analytic.
And the FDA approves drugs on non-analytical.
Well, that is true. That is
Right?
true.
It's
certainly
and when the
bolsters.
patients sit in front of us, we use a lot of non-analytical decisions
No.
to make those, I mean, you do. You don't say. I mean, we have PALA-1 is a non-analytic subset.
But if I worked for the sponsor, I'd say, look, I told you that olaparib worked in HRD test negative patients, and I told you that PALA-1 was a spurious result.
Yeah, maybe.
I would believe that.
What?
I think that's true.
I think that's true too. But that doesn't mean it's going to get olaparib approved in the HRD test negative.
Unfortunately not.
Unfortunately not. Any comments, Angeles or Brian?
Oh, well, I think it's been really well hashed out. I don't have a lot more comments. I think that, bigger picture, things move very quickly now. And you
That's
used to
right.
be able to wait until the control arm read out from another
Did it?
study before you develop something new. So I think it goes to clinical trial design. We have to have adaptable trial designs that can be fluid along with the information that we're gathering from new trials.
And let me throw
And I'm
this
not
out.
even sure how to do that.
And let me throw this out there. That's why the FDA allows us to modify the statistical analytic plan while the trial is ongoing. Now, ideally, you would analyze it before it stops enrollment, but even then, we've done that as long as the data remains, blinded. So I think we have to think more about adapting the endpoints as the data becomes available. We can't change the patients that we enrolled. We can't change the arms that we studied, but we can change the way that we did this. And if maybe this could have been amended while it was ongoing, and we'll have to see what DuoE shows. Go ahead.
And just a case in point here, we're still developing trials based on designs or traditional trial designs from 10 years ago. We need to start being transformational. How do we need to be
Bingo.
thinking about developing trials for 2030, 2040?
All right, well, speaking of trials that are transformational, and speaking of a trial that you're heavily involved with, Angeles, go ahead here, Tom.
Well, we just want to congratulate Dr. Secord for fully enrolling FLAURA5, GOG 3035 with Origovimab. That's been also a lesson in persistence, so thank you for doing that. I know there have been many iterations, but we're really excited to see the data. So
mine. Thank you, all of you, for participating. And also, just want to highlight, this was truly a global effort, and the sites in South Korea enrolled on the most studies, or the most patients on this study. So it just shows case in point how we have to work across all these different countries and have these synergistic collaborative partnerships.
I'm going to move on to something that's not very controversial at all, HIPEC. All right. So literally, when you go around and do wordsmithing with this and ask people their reactions, these are the kinds of reactions I get. It's a cure. It's a placebo. It's hope. It's a hoax. In fact, I wrote an article that was entitled that probably about 8 years ago because of the lack of randomized data that was out there. So you can see how polar this particular subject can be. And what really reignited the interest in this was the Vandriel data that came out 5 years ago that looked at doing the HIPEC therapy given in an interval cytoreductive environment. So that was important, caught us a little bit off guard in terms of people saw the recurrence-free survival there. Not all that great, 3.5 months, but then look at the median overall survival, almost a year. And so people were really moved by that. There's, certainly no study's perfect. Some people criticize it could be a type 1 error, could it be this, could it be that. Relatively small data set, 245 patients, so it's not very big, but the data has held up. And so Aronson presented the data this year at ASCO, looking at the 10-year follow-up, recurrence-free survival. You can see the hazard ratio is 0.63. Overall survival, 10-year follow-up, 0.7, holding with fairly large difference there in the medians as well. So this is obviously very significant data. So where do we stand with this? Well, this is looking at subgroup analysis from the study that tried to get an understanding of are there different subgroups molecularly that benefit more or less. And you can see here, based on very small numbers, they said that those patients with BRCA mutations, maybe not in this subgroup. There's other sensitivity analysis I'll show you that'll challenge that.
Can I just interrupt you there? I thought
Yeah.
intraperitoneal chemotherapy worked better in BRCA.
It works well. Everything works well in BRCA.
Oh, but not HIPEC.
If you're well,
Not HIPEC.
let's look at other sensitivity analysis. So I think that, this is data that was presented at IGCS this past year in New York. And there were two presentations at that meeting that were important. This is one of them. And this is, they looked at the role of HIPEC, both in primary and interval cytoreduction. The overall study was negative because they had a fair number of patients that were actually in the primary subgroup, perhaps obscuring any benefit in the interval group. So when you look at it and break it out, you actually see that in the group that received interval cytoreduction, the hazard ratio was 0.6 in terms of PFS and 0.53 in terms of OS. Now these numbers, again, are getting small. You're starting to select groups out, so you have to be a little cautious there. It was unclear as to whether this was a pre-planned analysis. It seemed like they went back and looked at it there after that happened. And then we had data that came out from the KGOG that performed a neoadjuvant approach with primary endpoint being PFS and OS. And here you can see the PFS is significantly different, 0.61, statistically significant. in terms of what was seen there, and pretty big difference on the median as well. As you look at the shape of those curves, and overall survival, very impressive hazard ratio of 0.31. Again, looking at it in an interval fashion. So I think we've learned some things. This is looking at other issues here, and here, Dr. Monk, you see that the BRCA did not affect the positive results. So, you know, again, still small numbers on this, but you have two small data sets disagreeing there. Something to keep in mind. At ASCO this year, we had more data that came out, and this is the CHIPPER trial that looked at the use of this in platinum-sensitive disease. So in terms of looking at it, at mostly all platinum-sensitive, these were first recurrence patients at first relapse, and they had to have a platinum-free interval of greater than or equal to 6 months. Again, they did this with interval, and then high-PEC versus no high-PEC. And I'll point out also there's differences in the dose. So Vandriel 100s, cisplatin here was 75. The temperatures have been slightly different, some 39 to 40, others 41. And so there's differences between some of these studies, but here you see the OS, which was the primary endpoint, has a ratio of 0.69, gets up towards 1, but does not cross 1, statistically significant. So interesting data in terms of looking at that, and that brings us to our current trial that looks at high-PEC in those at the time of interval cytoreduction, followed by niraparib maintenance therapy in those with stage 3 and 4 ovarian cancer. So we're really excited about having this trial open, and we'll see how that comes out. I'm excited because the logistics of this trial, I think, were well done in the sense that we're going to randomize after the surgery to get away from the surgical bias that we saw perhaps in Vandriel, where there was more upper abdominal surgery in the high-PEC group and maybe more of an effort to get those patients optimally cytoreduced. So I think, you know, the only question here is, you know, niraparib in everyone. What does everyone think about that? What is the role of Bev? The great equalizer that's not in here. So, you know, in clinical practice, would that change things? So I think even this trial will have some questions at the end, but an important trial for us to conduct.
So Brian, how important is it to have the same maintenance treatment in both arms of a high-PEC study? So if you look at the Vandriel and these others, and maybe it just balances out in randomization, so it doesn't matter. But that's why this trial, with you see the leaders there, Zavanovich and Randall, that's why they did this, to take that argument off the table. Also, really to standardize the high-PEC and to require training in high-PEC. Do you think that randomization, Brian, is going to sort it out, or do you think you need to pre-specify it?
As far as the maintenance?
Yeah.
Yeah, no, I think it's appropriate to have more of a homogeneous, a constant maintenance, because of you don't want to create more controversy when you're analyzing these results. So I think, you know, whether or not you use niraparib maintenance therapy in your HRP population or not on a clinical trial, I think it's reasonable to consider.
Yeah, so this idea of the HRD test negative, to your point, Tom, part maintenance, if the audience had not heard, the Athena Mono study got a complete response letter, meaning that their application was rejected. The PDUFA date was next week, and the FDA said, you know, because of all this survival controversy, and since the Athena Mono survival data are not mature, we're going to not give Athena Mono the FDA approval that they had requested. But for now, Dave, you know, niraparib is still there. So is this a reasonable control arm? Because the niraparib FDA approval, in their infinite wisdom, it's approved therapy. So I get it, Bev is an issue, but this is an approved therapy, so.
This is absolutely I think we really need to keep consistent within our maintenance therapies for all those biases. And let's remember, this is our only upfront first-line therapy treatment trial
That's
that
open.
we have. That's
Yeah.
open. So this is obviously a high priority, very controversial subject, clearly an unanswered. So I chaired the presentation, I forget what it's called,
Session.
Thank you, to cut to cure or something. But, we walked away from that with a question of, this is an unanswered question. And we need to have better design trials like HOT.
Yeah. And, to randomize, just to take out the surgeon's bias, that costs money, right? Because you have to activate the HIPEC team, and
Yeah.
there's things about that people don't think about. But we're doing it the right
we're
way
doing it.
here. It's being done the right way. The other thing I pointed out, different doses, different temperatures. We're going at the high dose and the high temp. So that will not be a criticism that we didn't give it the most aggressive way. So I think that's another good aspect of the
So,
HOT
Tom,
trial.
thank you. Excellent. On time.
Oh, we're not done yet.
Oh, we're not done?
That'll be late, don't worry, no. So platinum-sensitive disease.
Oh, we're on time. No, we're not.
I'll be very quick. So we've heard about the transformational effect of antibody-drug conjugates, and in platinum-sensitive, my role is to bring out the data or the trials that we have ongoing in this area. So we've been very excited about the apitegromab, rilsodotin, NaPi2b antibody-drug conjugate. As you know, the phase 1b and then UPLIFT was conducted and is fully enrolled. Just 2 days ago, we received the letter that there's a partial clinical hold on these trials that are here, the UPGRADE, which is looking at the UpRi plus Carbo, and the UpRi plus Bev cohorts that will be on clinical hold now because of 5 fatal hemorrhage events that occurred. So we're waiting on mitigation plan and then subsequent review by the regulatory agencies, and that puts up next in the same peril. So again, clinical hold for this as we move forward. Dr. Moore went over all the Mirasol data and so forth, but there are other opportunities to extend this, and these are in platinum-sensitive patients. So this is actually looking at using this as a single arm, using mirvetuximab in these platinum-sensitive patients that have had 2+ prior systemic therapies, so at least 2 rounds of platinum prior, and also the opportunity to build on Dr. O'Malley's data of combining this with bevacizumab in this platinum-sensitive setting as well. And I believe that takes me to the end.